I'm Dr. T, and welcome to my office. Today I'd like to talk about the central dogma of molecular biology. Okay, what on earth is that? I'm guessing you're asking. So the idea here is that we're looking at how information flows within cells. Cells can take information and store it really in kind of two forms, DNA or RNA. And you can also imagine that information is stored within proteins. But there's a bit of a caveat with that storage that I'll get to here in a second. So cells can take information stored on DNA and copy the DNA, essentially making two copies of that information. This is known as replication, and anytime a cell is going to divide, it's going to have to replicate its DNA. That way, each of the daughter cells gets a copy. At least that's how it normally works. Now, you can take that information on DNA and you want to do something useful with it. Simply copying information for information's sake is not that helpful. So what we can do is we can take, and by we I mean our cells, can take the information on DNA and copy it, but copy it so that the new product is not another set of DNA, but is now RNA. This is called transcription, so kind of like transcribing a video or something. Now you have the same information, but it's now in a slightly different type of molecule. Now this RNA could decide to do whatever it needs to do, or it could be messenger RNA, which after a bit of editing and post-production, it can take the information on the messenger RNA and use that information to construct proteins. This is known as translation. You're translating the information in the form of nucleic acids, which is a bonding or hydrogen bonding base pairing style information, to the language of proteins, which is shape, specifically the order of amino acids on a protein. Okay, now we've got a couple other things we can do. We can do reverse transcription. That's where you take RNA and turn that into DNA. And by we, I should not mean ourselves. Humans don't do this. Viruses, however, that like to infect us humans do. Additionally, once again, more of a virus thing, the RNA can be copied as well. So you can take RNA and copy it and make more RNA. Once again, not something we usually do. That's more of a virus thing. Yay, viruses. Not yay. Now, one thing you'll notice that I've left off is proteins going back to mRNA. Once the information has been translated into the protein, there's no going back. It can't be done. Well, I should say that we have not found a case, and we, as in science as a whole, have not observed a case where information from a protein is turning back into mRNA or some other form of RNA, nor do we know of a mechanism whereby that could occur. So, to the best of everyone's knowledge, it can't do it. That does not happen. Well, at least without actually sequencing an RNA and using computers and whatnot, but, that's a completely different kettle of fish. So in later videos, I'll talk more about the various parts of the central dogma, focusing mostly on those that we as humans can do. With that said, have a wonderful day.